A big welcome to the people who have already joined the session. The session is about can AI predict patient outcomes in ovarian cancer? With me today will be Dr. Anna Laury. I will also shortly introduce her as well. The idea of this presentation is to show you or showcase to you a research project that has been executed some time ago using the Aiforia platform, and how the different steps have gone, what kind of outcomes has been received, and quite an innovative work that has been done by Dr. Anna Laury. A little bit about Aiforia as a company. So we are a medical software company. We started quite many years ago. We have multiple different types of products at this point of time. The products that at least I would like to quickly highlight is our research product, which is the Aiforia Create, as we call it, and the Aiforia Clinical Solution. At this point of time, we have been deploying our solution in more than 50 countries with over 5,000 users of different products that we have launched in the market. We are an AI company. That's our main focus. We are an AI company mostly in the field of pathology, but we are also working on other fields as well. As briefly said previously, the clinical diagnostic sector is our ever-growing largest growing sector at this point of time, with so many other hospitals adopting digital pathology and also adopting the fact that AI along with the digital pathology needs to be or should be included in the day-to-day practice. In addition to that, research. Many pathologists in today's world, because they have a little bit more extra time because of AI, and not in all cases, but in those places where AI adoption, and in addition to the digital pathology, wherever it has happened, more time for doing innovative research has come into the limelight. Yeah, so that's why the Aiforia Create as a great tool is also available for the same very clinical practice researchers who would like to get their hands dirty with making some innovative algorithms and then bringing it to the clinical side. Yeah. Okay, that's a short wrap up from my side. The host, I forgot to introduce myself. So I'm Darshan Kumar. I'm heading a team of scientists here as a director of field application science and the Helsinki headquarters. At this point of time, the company has also offices in some other parts of the world like US and France and UK. But yeah, a big welcome to all of you. And I would like to really welcome Anna. Anna is a docent at the Helsinki University, and she has a medical degree from the University of California in Los Angeles. And she's a pathologist training. She is has had her training also from the Brigham and Women's Hospital. Her clinical interests include prenatal pathology, and her research focus is on development and progression of tubulo-ovarian serous carcinoma. And this specific research that Dr. Anna is going to talk about is on the serous carcinoma, more on the prognostic side. So it's a really interesting thing for me personally, because it's not just the predictive part, but also looking beyond what you can see with your eyes. Yeah. With that, I would like to give the floor to Anna, and thanks so much for joining, Anna.
Thank you, Darshan, for the introduction, and thanks to Aiforia for giving me the opportunity to talk. So the topic is, can AI predict outcome in high-grade serous carcinoma? And a little more specifically, this slightly off-the-wall project is about AI-directed spatial transcriptomics of specific areas of high-grade serous carcinoma. So just briefly, this is what I'm going to talk about. A brief history of high-grade serous carcinoma, just to orient you as to why we started this project. Very briefly mention AI for pathology, and then, of course, how we made this particular model that we used in this project, and then review the spatial transcriptomics that we did later. Okay, so as most pathologists know, but sometimes we forget, the microscope is really a rather recent innovation in the field of medical diagnostics, and it really didn't become popularized until the mid-1800s. When the microscope really began coming into use, we began to recognize that disease was more than just the organ that it arises in. It comes from cells and tissues. The H&E was developed in the mid-1800s, and that's, of course, when pathology began to develop as a discipline of its own, and not simply a subset of, for example, surgery. Then the early 1900s to Up until around 1990, we really just developed our understanding of disease, and this is when chemotherapy started coming into the clinical side. And this is also when the very first serum marker for high-grade serous carcinoma, CA125, was reported in the mid-80s. Right around the year 2000 and somewhat after, this is when genomics came into play. So first we had histology, then we had chemotherapy, and then we had genomics. The Human Genome Project was published, and the WHO Blue Books began to include genetic information. As far as ovarian cancer specifically, we started to really further refine our diagnostic capabilities, and specifically for high-grade serous carcinoma, it became a more specific diagnosis. Some things were excluded, and other things were brought into the fold of high-grade serous, and P53 was really recognized as the defining characteristic of this tumor type. And then finally, in the 2010s, we had the very first targeted therapy for this tumor type, and that is, of course, the PARP inhibitor, which exploits deficiencies in DNA repair in about half of these tumors. So where are we now in 2025? Well, we're still using that H&E from the mid-1800s. We're still essentially using that same chemotherapy from the 1970s, and we're using the biomarker from the 1980s. So that really kind of tells you that in the end, we haven't gotten all that far with high-grade serous carcinoma. Although we understand that it starts in the fallopian tube, we really haven't figured out too much more about it beyond the PARP inhibitor story. So it's still by far the most common so-called ovarian cancer. But while it's the 11th most diagnosed tumor in women, it's the 5th leading cause of death. So it's a much more deadly cancer than some other types. The 5-year survival still hovers right around 50%, even after the introduction of PARP inhibitors. There's really no population level screening, although this has been attempted. And as I said before, we're still basing most of our diagnostic work on the H&E. And therapy resistance remains a problem. So how much has been done? Well, quite a lot, even though ovarian cancer, so-called ovarian cancer, does tend to be really underfunded when compared to other tumor types like breast cancer. Still, people are always actively working on this tumor type. And if you search, for example, biomarker, quote unquote, ovarian cancer in PubMed, you get quite a few hits, about 23,000, even with that search term. So people are attempting to find ways to identify patients and give them more specific treatments and give them more information about their likely disease course. So also at this time, the field of pathology is undergoing a really rapid change. So what used to be a very human-centered field with an individual person looking through a series of lenses and mirrors at a very, very thin piece of stained tissue has really turned into this field where we're getting tons and tons of data, both from the image of the slide itself, so scanned images like you would use in Aiforia, as well as all kinds of data coming from those really thin pieces of tissue that we put on the glass. And so AI, of course, is what we normally think of when we think of AI for pathology, convolutional neural networks, where we label something that we want to find in the slides, like blood vessels, lymphocytes, and tumor, and then train a neural network to identify that for us on a new slide that it hasn't seen before. Just to place where this project is that in the overall field of AI, or AI for pathology, even, of course, there are the generative models that are all the rage right now, and that includes mostly multimodal models, so using different types of input. For example, these types of models, you could give it a text prompt and it will give you an image result. Or, for example, you could put in a picture of serous carcinoma and it would give you a description of that image. Then there are the foundational models that are created using entirely unlabeled images and other information, and those really function by transfer learning. It's a little bit like a medical student going into pathology. They use all this information that they learned in medical school, and then they have to figure out, or we have to figure out how to teach them to apply that to the field of pathology. And then, of course, there's the traditional AI classification-based approaches, and then unsupervised methods that basically result in clustering. And so we used, of course, a supervised approach, which can be things like identifying cells, so like objects, or regions like areas of tumor versus normal. And our specific approach was weakly supervised, meaning we did annotate kind of general areas and we labeled them as favorable or unfavorable, but we did not label any objects or any particular findings within the H&E slides. So why did we do this? Well, clearly high-grade serous carcinoma needs some additional help, right? We need to keep studying it because we really don't understand very much about these tumors and how they respond to chemotherapy and why some people do well and some people don't. And the histology does look different. These tumors don't all look the same. There are a lot of different morphological patterns that we can see. So we can see the classic patterns, which are shown here on the left, where it's the papillary and micropapillary morphology. And then there's this other set of morphology patterns that were described quite a few years ago now as the set pattern. So solid, which you can see in the middle, intermetroid on the right, and transitional there on the bottom. And those were noted to be associated in particular with patients with pathogenic variants of BRCA, both 1 and 2, but especially 1. However, as this image shows, those descriptions really haven't turned out to be very useful in practice because they're really not specific or sensitive enough, even though it has been shown repeatedly that they are associated with BRCA or other homologous recombination deficiencies. So you can see here on the bottom left of this image, really classic papillary architecture. Then towards the middle, you can see really solid tumor growth. And then on the right, I don't think anyone would argue with calling that a transitional pattern. So you can see even in this one tumor fitting in this one image, you can see all three pattern types. But that does tell us that there is likely to be something there, or at least it told us at the beginning of this project. We have these patients that respond differently to chemotherapy, and then we have all these morphological differences that kind of are associated with certain things, but not in a very, not in a way that we can yet use clinically. And so this is what we wanted to do. We wanted to be able to take slides from patients, put it through this model, and have the AI model tell us if this patient is likely to respond to chemotherapy, so have a long interval before recurrence, or is unlikely to respond to chemotherapy and recur right away after the end of chemotherapy, because the top group is really unlikely to get a lot of benefit from current chemotherapy protocols, and we want to be able to identify them and appropriately counsel them on the likely disease course that they will have, but also work to find better treatments for these patients. And so this was our overall approach. We took a group of patients who had the same presentation, so late stage disease, which is how most of these people present, with tumor already all over the abdomen, who received the same treatment. So in our case, at least 6 cycles of platinum-based chemotherapy, but despite the same presentation and same treatment, they had very different outcomes. So half of the patients had a very favorable outcome, meaning that after they completed their first cycle of chemotherapy, they went 18 months before they had any sign of disease progression. And the other group of patients had an unfavorable outcome. So after the completion of the first line of chemotherapy, they recurred within 6 months or less. So we wanted to compare these two groups and see if the AI model could find anything in their histology that was helpful to tell them apart that we couldn't already see with our eyes. And so we did this iterative process to create our classification model, first tumor detection, then outcome prediction, then high confidence region filtration, and finally we trained a classification model. And so just actually, I was supposed to say that here, but these are the 3 models that we trained. Tumor detection, so just annotating for tumor. Outcome prediction, so that's where we annotated tumor and then labeled it either favorable or unfavorable outcome. Then with the results of the outcome prediction, we filtered it for high confidence regions, annotated those, and that trained model was able to classify slides. So here, just in cartoon form, because it tends to be a little bit confusing. First, just tumor detection, which is pretty basic. And then outcome classification, which just annotating the tumors for outcome and training models really wasn't that successful. And all of the test set slides just tended to have areas of both, and we couldn't really use that data to classify the test set slides. So then we decided to say, okay, let's take these area the arrows should be over here, but apply a high confidence filter to these results. And then those results, which are much less area on the slide and will only be either favorable or unfavorable outcome, annotate these areas here and train a model. And that trained model annotated on the high confidence areas could then classify our test set. So 18 of 22 slides were correctly classified, which I will admit is not super amazing, but it is a whole lot better than we expected the model to do. And so we were pretty surprised and happy about that result. So then we thought, okay, what are those areas? So to answer this question, we decided to make a TMA. And in that TMA, we had 4 different types of tissue. So from the poor outcome or unfavorable outcome tumors, we had the areas identified by the model, and then areas not identified by the model, which we called background tumor. And then the same for the favorable outcome. Both areas identified by the model as being predictive, and then so-called background tumor areas that were not predictive according to the model. And these are the TMAs that we made, 4 tiny TMAs. And we made them that size in order to do spatial transcriptomics. So as I'm sure all of you know, spatial transcriptomics just means basically, in this case, the protein coding RNA content of the individual cell or tissue that you are looking at. So while DNA changes are relatively static for each individual cell, the RNA is, of course, a very dynamic it's dynamic and it changes throughout the life cycle of the cell and the tissue, and it can kind of give you a better idea of what's going on within the tumor and between the tumor and the stroma at the time that you do your measurements. And so we, as you could probably guess by the size of the TMAs, we first started with the Visium for FFPE system. And here you put the piece of tissue in this tiny little capture area that has all these little dots on it. The tissue sits there, and then it goes through various treatments to release and anneal all the pieces. And in the end, a spatial barcode is attached to all of the little RNA fragments, which are then copied and sequenced. And this is a kind of an interesting system because it really works for fishing expeditions. You don't have to have any idea what you're looking for, though, of course, it certainly works better if you do have an idea. But because it has about 18,000 protein coding genes in its set, you can answer a lot of questions that you didn't even have to start with, depending, of course, on the type of tissue that you use. So what did we find? Well, if you look at the UMAP, which is just, of course, a dimension reduction to sort of try to visualize for a human all of this incredible amounts of data that we get from even an experiment like this. It turns out that each of these clusters that you see here in the UMAP represents one patient, which is not too surprising because we already know that high-grade serous carcinoma is extremely heterogeneous, both within patients. So like tumors at different sites are not necessarily the same, but then, of course, between patients. So patient A and patient B are likely to have a fairly significant transcriptomic profile. But it was interesting to note that the background and the high confidence regions, so the high confidence or the AI identified regions, did seem to be rather discrete within each patient. And so our brilliant bio-IT colleague over here, Shuyu, did some various types of analyses, including spot distance calculation. We kind of try to measure how similar and dissimilar all these spots are. And essentially what she found was that the background spots from favorable and unfavorable outcome are more alike than the high confidence or AI identified spots from background and high confidence. From high confidence areas, sorry. So short and long are more different in the high confidence areas than they are in the background areas. And of course, the things that are more different from each other are easier to tell apart and are more predictive. So that was like very exciting. And that's when I started to think, maybe the model really did do something here. So then we just decided to do a little more AI, but not image analysis AI. And so Shuyu trained quite a number of models just to see, are the transcriptomic profiles from the high confidence AI identified regions more predictive of outcome? And no matter what type of model she trained, they were always slightly more predictive. So you can see here for the GLM and the other type of convolutional neural network, you can see that the blue, or that's the transcriptome from the background regions of tumor, are basically at chance in both of these. But the red one, while not an amazing result, it's clearly much better at predicting outcome than the background blue line, which was reassuring. Then, of course, we also did pathway analysis, and this was interesting and perhaps not entirely unexpected, but the poor outcome, high confidence regions tend to have kind of enrichment for stress response types of pathways, whereas the high confidence from the favorable outcome tumors tended to have enrichment for DNA repair pathways, a large number of them, not just homologous recombination, but really a large number of pathways, which was kind of interesting. And there were certainly more significant results in the high confidence regions than in the background regions. Looking at a protein-protein interaction map, we found that June really seemed to be central to a lot of these enriched transcripts for various proteins. Okay, so then the reason that we identified one particular thing in particular here being June in this example is we wanted to see, okay, we can see it here in the spatial transcriptomics, we can see it in the data, we can see it when we visualize the expression on the individual spots, because of course in Visium part of the idea is that you can see the data overlaid on the H&E, and you can see quite clearly here that in the high confidence for short outcome areas, there's more June than in this high confidence for long outcome, which is consistent with the data showing that there's more June expression in patients that do not do well with standard chemotherapy. So then we wanted to say, okay, if we're thinking that we would want to make this clinically applicable, we want to use a method that you could use in the regular pathology lab, because of course nobody's going to be doing spatial transcriptomics on all their tumors. That's kind of, that's not going to happen. So we did RNA-ish for June. And I think these pictures, even if they're kind of small, you can clearly see that the tissue with the most June expression, or the most RNA-ish signal, is from a high confidence short area, all this brown stuff here. And the high confidence for long outcome area really has very little. And the background areas each have some. Maybe short has a little bit more than long, but there's more brown over here on the right than on the bottom left, which is high confidence long. So this is confirming both the visual transcriptomic results and the numbers. Then, of course, one of the most interesting things about this is that you can do cell-cell interactions. And so one thing that we found that is something you can't see just looking at the slide is that a specific type of tumor cells, so tumor cells, the name of it doesn't really matter, but this is a tumor subset that's really enriched for DNA repair pathway activity. So those was seen in high confidence long tumors, and associated with its EOC-C5, or the specific tumor subset, there was a notable lack of macrophages and plasma cells, which is kind of interesting. The high confidence short, of course, has cells with similar activation, but they are associated, the macrophages are either there or not there. So that was an interesting cell-cell interaction information that we could find from this that we weren't able to see with our own eyes, though perhaps the model was. So then we did also do some nanostring on these same TMAs. And of course, for nanostring, you have to select a specific area using their area of illumination. And then all of the transcript data from this specific area in the box is going to get pooled. So you just get the one result for each area that you do. And I'm not going to really talk about this very much because this isn't really the point of this presentation, but just to say that the results from geomics correlated very well with those from Visium, even though the techniques really are quite different. Even though they're both transcriptomics platforms, the techniques are different and the type of data that you get is different. But overall, the whole transcriptome DEGs were very well correlated overall in selected genes of interest. And then, interestingly, there are some transcripts are identified only in the geomics data and not in the Visium data, although they are listed in the panel for Visium as well. And so we did some RNA-ish on that as well. And again, the findings were confirmed by the RNA-ish on tissue. So in summary, we have with Aiforia created an AI model that discriminates extremes in outcome in high-grade serous carcinoma, which is something that humans really can't do on our own. It identified morphologic features that are novel, perhaps. These AI-identified regions, or the high-confidence regions, do have distinct transcriptomic profiles, and they are more predictive of outcome than other tumor regions. And then two spatial profiling techniques can identify novel relationships, and the results do seem to stay fairly consistent across platforms, which is reassuring. And so is there more than meets the eye here in the histology of high-grade serous carcinoma? And apparently you viewers thought that, yes, there is more than meets the eye here. And I now agree with you after going through this project. And this was the team that did the work with me. Of course, it takes a lot of people to do all of this. And that's what I have today. And if there are any questions, I'm happy to try and answer them.
The first one is it mandatory that all the H&E sections are cut and stained at the same time and at the same lab? If not, is it possible to collect sample sections from different hospitals? Question mark. Would you want to answer that, Anna?
Sure, sure, yes. So, I mean, that is a great question for like a clinical test. I think so. The slides that we used in this project were all from the same hospital, although they were not all cut and stained at the same time. They had a range of about 8 years between the first and the last, so they don't need to be cut and stained at the same time. These were all stained at the same lab. We have tried to expand this to test it from on slides from another hospital, and I will honestly tell you that it did not work very well. But then we looked at it and we saw that that hospital actually has remarkably good outcomes, at least in the cases that we were able to use. And so I think that we weren't actually able to our model did not generalize well without any retraining, and then we couldn't really add enough cases to have both a retraining and a test set. So still is a little bit of an open question. So of course the answer should be no, but I'm not entirely sure. Perhaps we just got really lucky with this model and it's overtrained and overfit specifically to our data. But I would hope that we could also generalize it if we had enough slides from another institution.
Thanks, Anna. We have another question coming in. Can you recognize the different high confidence areas? Question mark.
I guess that means histologically on the slide.
Yeah.
I mean, honestly, not really. They do tend to be more solid for the favorable outcome, high confidence regions, and more kind of traditionally papillary for the unfavorable outcome, high confidence regions, but it's not distinct enough that I can separate them, for example, by myself. But there do seem to be some general differences, but it's not so precise that you could use it as such without help.
Okay, thanks. Another one, are prognostic AI models becoming more popular these days, and who are they most beneficial to? Maybe I can answer that. So at least this research, in addition to other research that I have been seeing also and also helping, they are becoming quite popular. That's true. That's also because the AI engines are able to deduce this kind of an information from not so simple H&E, because they have a much deeper story to say. But we have been seeing now research on breast cancer, chemotherapy response, on prostate cancer survival, on colon recurrence-free survival. So they are becoming more and more common at this point of time from our existing research project or clients. Okay, there's another one from in front of me. How can this AI tool translate into the clinical for patient prognostics? Is it feasible with just one cohort set to state that this model can predict outcome in these patients. It has not used an external validation cohort, question mark. Anna, would you like to address that?
Yeah, so of course, this person is completely right. Yes, this is not, I would not say that this is a functional clinical tool for use really in any patient population. I think this is really a small proof of concept study. Is this something that is possible? And then it was possible, and then we tried to prove some type of biological meaningfulness. I'm not sure if that's a word, but this is really just a proof of concept study, I think. I totally agree that if you wanted to try to use it as a clinical tool, it would need to be trained and tested with multiple external cohorts from multiple places.
There's another question. Have you tried training a model with spatial transcriptomics data as such, I guess? Anna, maybe towards you, that
Yeah,
question.
I guess that's for me. We have not tried training that, though I know that people do that. And they even actually have been attempts to eliminate the H&E or the slide altogether and just use spatial transcriptomics to predict or say what would be there or what will be there on the slide. But we have not done that with this data, although that's an interesting idea.
Okay, thanks. Another one, did you do any MIF study of the TMAs?
Does that mean like multiplex immunofluorescence? I
It
assume
looks like so, yeah.
that was that was in our plan, although we haven't actually done that yet. We do still have some tissue left, and we're kind of thinking about how to use the rest of it. And that would make beautiful images, I'm sure. So, but no, we haven't for these TMAs, we have not done that yet.
Then I would like to thank Anna for this session, for this presentation.